In a previous lecture, I described the way in which glucose is broken down, but glucose is not the only sugar that our cells need to deal with. In this lecture, I'll talk about the metabolism of other sugars. I'll discuss the process of gluconeogenesis, and that's the way that cells make glucose that they can use. And then last, I'll talk about the phenomenon of reciprocal regulation, the way cells control sugar metabolism. Now, cells have a lot of different sugars that they get besides glucose, so it's important that we understand something about what they do. In this pathway, we see the metabolism of a sugar known as galactose. Now, we get galactose in our diet fairly regularly if we eat dairy products because galactose comes from lactose, which is known as milk sugar. So our cells have to be able to metabolize galactose. To metabolize galactose, the metabolism is actually shown on the screen. I'm going to step you through it, and it looks a little complicated, but it's actually fairly simple. The reactions of galactose basically involve its conversion into an intermediate in glycolysis. Now, this starts with an enzyme that has a mouthful of a name, galactose 1-phosphate uridyl transferase. Hopefully, the reaction is simpler than the name of the enzyme is. The reaction catalyzed by this enzyme takes galactose 1-phosphate on the upper left and combines it with UDP glucose on the lower left. And what it's doing is essentially swapping the galactose for the glucose. So we see when this process happens that we start with galactose 1-phosphate and we end up with UDP galactose. UDP glucose releases a glucose 1-phosphate. So because of that, we now have glucose 1-phosphate that's free. Now, glucose 1-phosphate is relatively easy to get into glycolysis because there's an easy enzymatic conversion of glucose 1-phosphate to glucose 6-phosphate, and we get into glycolysis. This means that we've converted part of the molecules shown on the screen into glycolytic intermediates. The galactose in the UDP, however, has to also be converted into an intermediate. And that happens in the next reaction, which is catalyzed by UDP galactose 4-epimerase. Again, a mouthful of a name, but the reaction is quite simple. The galactose at the end of the UDP is converted into a glucose. That reaction means that we've now gotten to a point where we have made our starting material. UDP glucose on the right, UDP glucose on the left. The UDP glucose on the left now continues the process, metabolizing more lactose. So in this way, all galactose can ultimately be converted into glycolytic intermediates. Now, other sugars get metabolized. I've mentioned lactose. Lactose is milk sugar, and it's metabolized in a very simple way. There's an enzyme called lactase, and lactase breaks lactose into its constituent sugars, which are galactose and glucose. You've seen how galactose is metabolized. Now, lactase is an important enzyme because in some adults, the reduced quantities of this enzyme leads to a phenomenon known as lactose intolerance. And lactose intolerance, if you've ever had to deal with it, is pretty discomforting because it causes the bacteria in the stomach to be able to produce a lot of gas because they're working on lactose, not galactose and glucose. Now, another very important consideration, particularly in places where there's high fructose corn syrup, is a reaction I'm going to show you on the screen here. Now, high fructose corn syrup is produced in some places, primarily in the US, from corn. And high fructose corn syrup has, as its name suggests, high quantities of fructose within it. It has greater than 50% fructose. So if you eat sucrose, for example, you get fructose and glucose in about equal quantities. If you eat high fructose corn syrup, the balance is shifted in favor of fructose. Now, what I'm going to tell you here relates more to sugar metabolism in general than just fructose, but I use fructose as an example to show you why this could be problematic. Now, besides the enzymes in glycolysis that I've talked about, there are other enzymes that handle other sugars. So, for example, the enzyme fructokinase that you see on the screen converts fructose into an intermediate called fructose 1-phosphate. Now, that's a fairly simple reaction. There's simply the addition of a phosphate onto fructose using the energy of ATP. That's very much like we saw the hexokinase reaction, although it uses fructose and it's putting phosphate in a different place. Nonetheless, a simple reaction. Fructose 1-phosphate is the target of another enzyme called fructoaldolase. And what fructoaldolase does is it converts fructose 1-phosphate into 2 molecules of 3 carbons each. One molecule is called dihydroxyacetone phosphate. You remember that from glycolysis. The other molecule is called glyceraldehyde. And you learn part of that in glycolysis in the form of glyceraldehyde 3-phosphate. Well, the only difference between glyceraldehyde and glyceraldehyde 3-phosphate is the 3-phosphate. Cells can easily convert glyceraldehyde into glyceraldehyde 3-phosphate. When it does that, the end product is 2 molecules from glycolysis. So I said something was wrong with fructose metabolism. This looks like we're simply feeding glycolysis like we were before. Why would there be a problem? Well, we don't know the answer to that question, but I have an idea about it that I want to share with you here. If we look at glycolysis, what I've shown are the first few reactions in glycolysis. Glucose going to glucose 6-phosphate catalyzed by hexokinase. So in each case, the enzyme that does the reaction is shown in orange and the molecule is shown in green. Now, if we consider what I just described, which is the molecule involving fructokinase and fructoaldolase, we've bypassed the first set of reactions in glycolysis. Why was that important? We just produced glycolysis intermediates. The reason that's important is if you remember from glycolysis, we bypassed 2 enzymes that play roles in regulating glycolysis. Now, Houston, we might have a problem. Because what will happen is without regulation, this pathway that's feeding it can make abundant intermediates of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate under conditions where the cell might not normally do that. Now, yes, it's the case that this reaction might occur in the consumption of too much sugar in the first place, so it's a good reason perhaps not to drink too much sugar. But now, if we're starting to feed glycolysis when it normally would not be fed, here's what can happen. Okay? Bypass the regulatory enzyme. We get to the last half of glycolysis, and things are coming down the pike that wouldn't normally come down the pike. The cell would normally shut it off. But now this sugar metabolism is force-feeding glycolysis so that what could happen, again, this is my idea, this isn't anybody's proof, what could happen with this is we produce excess pyruvate. Why is excess pyruvate a problem? Well, pyruvate turns out to be a precursor of acetyl-CoA. And when you have plenty of energy and you have plenty of acetyl-CoA, what you make are fatty acids. And when you make fatty acids, you make fat. Some people think, and there's some pretty good evidence, that high fructose corn syrup correlates with obesity. So, my advice to you, be careful with the carbs that you're taking in. I want to spend some time now talking about the pathway of gluconeogenesis. This is the way that cells make glucose. And this pathway is very, very important, especially for a multicellular organism. Multicellular organisms have a fairly constant need for glucose. Our eyes need it pretty badly, our brain needs it, and they don't have ways of producing it. So maintaining glucose concentration is important. There's 2 ways that we can get glucose. We can get it from our diet, and we're not constantly eating, even though sometimes it might feel like that. And we can also get it from synthesizing it, and that's where gluconeogenesis comes in. So gluconeogenesis kind of fills in the gaps when we're not filling them in by filling our stomachs. It's important for cells to be able to control gluconeogenesis because it's the opposite of glycolysis, and we don't want to have a breakdown pathway going on at the same time as a synthesis pathway. So one of the ways in which this pathway is regulated is through localization. It turns out that in our body, gluconeogenesis doesn't even occur in every cell. It occurs primarily in our liver and our kidneys. And even within those cells, there is some localization of the enzymes, as we shall see. Now, the reactions of gluconeogenesis, there actually are 11 reactions of gluconeogenesis, and I'll talk about those, specifically the different ones, in this discussion. Last, I'll talk about regulation. So, let's get into it. The synthesis of glucose, as I said, was important for regulating blood glucose. And so we have reactions in gluconeogenesis that occur within a cell that actually does gluconeogenesis in the cytoplasm, in the mitochondrion, and the endoplasmic reticulum. You'll recall that all the reactions of glycolysis occurred in the cytoplasm. In the body, as I said, we make glucose in 2 primary organs, the liver and the kidney. And the liver of the 2 is pretty much the most important because it helps modulate all kinds of nutritional needs in our body. Now, in gluconeogenesis, there are 4 reactions that replace three reactions in glycolysis, and they're the ones that we will focus on for this talk. All the other reactions are simply the reverse of glycolysis reactions. There are 7 of them. In glycolysis, we had 10 reactions. In gluconeogenesis, we'll have 11. So to make a glucose has 6 carbons, we need 2 molecules of pyruvate because there's 3 carbons in each pyruvate. The reactions of gluconeogenesis are shown on the screen here, and this is a kind of a complicated pathway, so let's try to break it down a little bit. There are 7 enzymes, as I said, that are common between the two, and they're shown in dark green here. I won't talk about those because the reactions of gluconeogenesis that use these enzymes are simply running the reactions of glycolysis backwards. The enzymes of gluconeogenesis that are different from glycolysis, I want to spend a little bit of time talking about because they have some considerations for us. The first of these reactions that I'll talk about is the reaction catalyzed by the enzyme known as pyruvate carboxylase. You can see it on the lower right, and in this reaction, pyruvate is being converted into oxaloacetate. Well, what does that mean? Pyruvate has 3 carbons, and for this reaction, a carbon is actually added to pyruvate. That comes from the bicarbonate that you can see at the bottom of that image. Now, ATP energy is required in order to make this happen, and so we produce ADP in the process. These are the intermediates that we're talking about right here. Now, why is the cell doing this? Why is the cell making oxaloacetate? Why didn't it just reverse the reaction to go from pyruvate to phosphoenolpyruvate? Well, that was the big bang of glycolysis, and that reaction is essentially irreversible. The cell is having to take a 2-step around that, the first step of which we just now covered. The first step, using the enzyme pyruvate carboxylase, happens in the mitochondrion. The first step happens in the mitochondria. It's physically isolated from the reactions that are occurring in the cytoplasm. After that reaction occurs in the mitochondrion, the oxaloacetate that's produced is moved out to the cytoplasm. Now, here's the reaction that you just saw. The pyruvate carboxylase is an enzyme that uses a coenzyme. Now, coenzymes are important helpers for enzymes. And what they usually do is they have a specific function. The specific function of pyruvate carboxylase is to grab hold of a carbon and put it onto something. And that's what's happening here. The 3-carbon intermediate pyruvate is becoming the 4-carbon intermediate oxaloacetate. Biotin is helping pyruvate carboxylase to do this. Now, after that reaction of making oxaloacetate, the oxaloacetate, as I said, is moved out to the cytoplasm. And in the cytoplasm, there's an enzyme called phosphoenolpyruvate carboxykinase. Yeah, mouthful of a name. PEPCK is actually one of the enzymes that is regulated at the gene level, meaning that most of our cells of our body won't make this enzyme. And if they don't make this enzyme, then they can't do the next step in gluconeogenesis. So we can see how gene regulation helps to control a metabolic pathway. Cells that don't have PEPCK, as we call it, can't go through gluconeogenesis because they can't catalyze this reaction. But this enzyme is activated and made in liver and kidney cells where gluconeogenesis occurs. Well, let's look at the reaction. In the reaction that's happening right here, this reaction converts oxaloacetate, OAA, into phosphoenolpyruvate. And notice that it's using the triphosphate known as GTP to make it all happen. The GTP has the same energy as ATP. So in essence, in going from pyruvate back to PEP, we had to use 2 triphosphates to make that happen. That big bang was pretty hard to overcome. We've just seen the 2-step that cells have to go through to go from pyruvate back to PEP. The next steps will actually be quite easy for students to learn. And the reason is because they're simply the reversal of the reactions that were catalyzed in the glycolysis pathway. The very same enzymes run in the backwards direction go from PEP back to fructose 1,6-bisphosphate. At fructose 1,6-bisphosphate, however, there's a different enzyme that's used to make gluconeogenesis work. And there's a very good reason for that. The next reaction of gluconeogenesis is catalyzed not as the reverse of the PFK reaction, The first reason that that is not done is because PFK is catalyzing a reaction that is energetically favorable in the direction of glycolysis, and it's difficult to overcome that. So rather than have to go through a two-step, the cells are simply bypassing it, as we will see here. The second is that PFK is set up very well to regulate glycolysis, but it's useful to have a different enzyme to regulate gluconeogenesis, and we'll see how that happens in just a little bit. So the enzyme that is used in gluconeogenesis instead of the enzyme that's used in glycolysis, the enzyme in gluconeogenesis is called FBPase, as you see in the abbreviation, or its longer name is fructose 1,6-bisphosphatase. Now, this reaction is not the reverse of the reaction in glycolysis. And the reason it's not is because in glycolysis, the PFK enzyme required ATP to put the phosphate on. In the reverse reaction, all we're doing is clipping the phosphate off. We're not regenerating the ATP that was used in glycolysis. So the cell is actually cheating here again. The reason it's cheating, it's not completely reversing the reaction, even though the intermediate fructose 6-phosphate is still generated. What the cell's doing is it's using that high energy of the phosphate on position 1 to drive this reaction. And all it takes is water. To go from fructose 6-phosphate to the end of the gluconeogenesis pathway is now pretty easy. What happens is, first of all, the fructose 6-phosphate is converted back to the glucose 6-phosphate by a reversal of the glycolysis pathway, as we saw before. That leads us to glucose 6-phosphate that needs to be converted into glucose. And here the cell uses the same trick it used in converting fructose 1,6-bisphosphate into fructose 6-phosphate. It uses a different enzyme. The enzyme here is called G6Pase, or glucose 6-phosphatase, and the reaction that it's catalyzing is simply the clipping off of the phosphate from position 1. Notice again, this is not the reversal of the hexokinase pathway. The cheating that the cell is doing is it's not recreating the ATP that would be required for the reversal of the reaction. So the energy is realized by not regenerating that ATP. This reaction is another reaction that does not occur in the cytoplasm. This reaction occurs in the endoplasmic reticulum. So again, we see some sequestration that's happening, and we also sort of see why all those other reactions were occurring in the cytoplasm, and that was because many of them were using the same enzymes as glycolysis. Let's turn our attention now to thinking about regulation, because regulation is very important for making sure that these two pathways are not at loggerheads with each other. Glycolysis goes as follows. You start with glucose, 2 ADPs, 2 phosphates, and 2 NADs, and you get the products shown below. And in gluconeogenesis, we're reversing the process, and if you add up all the different pieces that I gave you in that pathway, we see that we start with 2 pyruvates, 2 NADHs, and then it takes 4 ATPs and 2 GTPs to make the glucose. Well, if you do the math, what you see that it takes more triphosphates to make glucose than you get out of glucose when you break it down. Well, that makes sense. There's no process that's 100% efficient, and this one isn't either. But the problem with that arises if we turn and we try to run both these pathways at the same time. Glycolysis makes pyruvate, pyruvate goes up to glucose, glucose goes through glycolysis, and we go in this circle. It's actually called a futile cycle, is what it's called. But this futile cycle does nothing but burn ATP and GTP. When you burn things, what happens? Heat and loss of essential energy that you need. So the futile cycle should be avoided, and that cells have interesting things set up to avoid that futile cycle. ATP and GTP get burned, as noted, but nothing but heat is produced. Now, if you're trying to generate heat, that could be useful, but usually you're not trying to generate heat. So if we look at the regulatory enzymes in glycolysis and gluconeogenesis, and you're seeing the gluconeogenesis ones for the first time here, we discover that many of these have reciprocal effects or opposite effects on one enzyme compared to the other. We see, for example, that PFK is activated by AMP. That makes sense. Cells need energy. They want to get PFK going. If cells need energy, they certainly don't want to have gluconeogenesis going because it's going to take energy to make glucose. Notice that FBPase gets turned off by AMP. Notice that PEPCK gets turned off by ADP. That's a lower energy indicator than ATP. And notice that pyruvate carboxylase is turned off by ADP. So low energy things are turning off gluconeogenesis things and turning on glycolysis. Hexokinase is turned off by glucose 6-phosphate. That may not be apparent, but the times when glucose 6-phosphate is high is when the cell has plenty of energy. PFK is turned off by a high energy compound, ATP. And pyruvate kinase, the Big Bang catalyzer, is also turned off by ATP. Gluconeogenesis, on the other hand, is activated by things that indicate high energy. Now, citrate is one of these things that indicates high energy. With citrate, the fructose bisphosphatase, FBPase, is activated. Another thing that indicates high energy is acetyl-CoA. Now, acetyl-CoA, you may remember, is one of those things that if it's produced in excess is going to be a problem, and you make fat when you have high energy, and if you have a lot of acetyl-CoA, high energy. So these high energy indicators are having opposite effects on glycolysis and gluconeogenesis. Now, there's a couple of things here that don't really have any relationship or any direct relationship to energy. The most important of these is the molecule called fructose 2,6-bisphosphate. I mentioned in the glycolysis talk that fructose 2,6-bisphosphate was an activator of PFK. And you'll notice there's 2 plus signs by it. It's a pretty strong activator of PFK. You'll notice also that fructose 2,6-bisphosphate has 2 minus signs next to FBPase. Those two enzymes catalyze equivalent reactions. In the glycolysis direction, we're going down from fructose 6-phosphate to fructose 1,6-bisphosphate. In the gluconeogenesis reaction, we're going up from fructose 1,6-bisphosphate back to fructose 6-phosphate. They're catalyzing opposite things. And notice that the regulator is having opposite effects on them. It's turning off the gluconeogenesis enzyme at the time it's turning on the glycolysis enzyme. This reciprocal regulation is a very important concept to understand in metabolic control. Now, one of the things about fructose 2,6-bisphosphate that I should mention is that fructose 2,6-bisphosphate is made upon insulin stimulation. When is insulin made? Well, insulin is made when the liver, for example, starts getting glucose from a meal. And you might think, well, with a meal, do I need to deal with all of that glucose? Should I be burning it like I'm burning it here? Well, it turns out that the liver's got a little different circumstance than the rest of the body. The liver has a lot of glucose, and glucose can be toxic. So glucose must be dealt with, and insulin is there to deal with glucose. What insulin does is it stimulates the uptake of glucose, but if you take up too much glucose, you have a problem. So cells have to deal with that excess of glucose. How do they deal with it? In 2 ways. One is by burning it with glycolysis, and the other is by storing it in the form of glycogen. Insulin stimulates both of those processes and results in the reduction of glucose concentration inside of cells. Now, fructose 2,6-bisphosphate is broken down by the opposite thing, and the opposite thing is glucagon stimulation. Glucagon says that there's too little glucose around, and the liver's got to worry about too little glucose because the liver is supplying glucose to the rest of the body. What is it doing? It favors gluconeogenesis. And what does gluconeogenesis do? It releases glucose for the body. So the opposite effects of insulin and glucagon control the synthesis of fructose 2,6-bisphosphate and help supply the body with its needs exactly as needed. In this discussion, I've gone through the metabolism of other sugars as well as the synthesis of glucose, and I've talked about the ways in which these pathways can be reciprocally regulated. By balancing the pathways as I've described here and using very simple concepts of reciprocal regulation, the cells in our body can use and produce glucose in the way that it needs.